Very nice, Eddie. And I have a question. The timing, it is only in adult cells or in the embryon development, this methylation of arginine plays an important role?
Well, here so far we have everything in tissue culture, and so we're lacking before we can publish this to have a good demonstration of the requirement of methylation in the embryo, which is what we work with. Wnt signaling, it works in all these tissue culture cells. That's where mostly is studied. So we would think that it will also happen in the embryo. Now, to study the very early embryo in the egg, that is quite difficult, and we're trying hard. But sure. This first rotation, there's a lot of yolk, and the multivesicular bodies get incorporated into the yolk itself, so that's very difficult. But this is taking place, at least in the gastrula stage on Wnt signaling. This we know. So we expect it to work in the embryo, because that's what we work with, really, the embryo. But yeah, so we're working on it.
Thank you.
Steven?
Hi, Ed. Here. So it's very interesting. The Wnt has a very pleiotropic role, like in development, and the knockouts are basically lethal, but the conditions you can study with the conditions, right? The conditional knockout. My question is like the role of Wnt in lysosome and butyrination. How can you dissect the participation of the Wnt in this process associated with the other roles of Wnt, for example, in several diseases? Do you think that the main role of Wnt being associated, for example, with the lethality and with some diseases is associated with their role in these endosomes and in this degradation of proteins?
Well, Wnt is involved in a huge number of diseases, but what we have done in embryos and in cultured cells, we've shown that you absolutely need the formation of the intraluminal vesicles to get any type of canonical Wnt signal. It's called canonical, not because we're here, but it's the beta-catenin dependent signaling. So for that, you absolutely need what's called the escort machinery. So we have shown that. On the other hand, well, there are so many diseases that are mutations in the escort machinery that it's a little bit difficult to untangle. But this methyl transferase, for example, is lethal at the third day in the mouse. It's completely, a very major enzyme. So,
Pablo?
Is there any other example, in addition to beta-catenin, whose degradation dependent on Wnt, regulated by Wnt, regulates a specific signaling pathway as opposed to just bulk degradation of proteins?
Well, yes. Well, we have studied this quite a bit. For example, if you take what's called TGF-beta signaling, which should have this protein SMAD4, know that when you have Wnt signaling, then that is much more active. So if you put, through this sequestration of GSK3, in the case, we have whole papers on that they're so prevalent, these phosphorylations, they affect at least the TGF-beta and the BMP pathway also. It's a very important problem about the isolation of individual signaling pathways. But the answer is yes. We think that it's involved in multiple pathways. But the transcriptional effects of Wnt are always through beta-catenin. So we've tried it in many different ways. If you remove beta-catenin, you don't get the transcriptional effects of Wnt, no? So, yeah.
Yep.
Yeah. No, you said that the two pathways of degradation are not independent, that you can have a switch from proteosome to autophagosome to proteosome. Is there any evidence of the contrary? I mean,
No, the answer would be no. We don't have any evidence in the other direction, but the evidence we have for saying that proteasome, it has to do with this protein SMAD4, for which we have very good antibodies. SMAD4 is cytosolic or nuclear, but when we put Wnt, it goes into these endosomes through this process of microautophagy, which is very neglected because it's the formation of these little vesicles. And it's it's just thought to happen and not even to be regulated. So what's important about our work is that the microautophagy is regulated, no? As opposed to macroautophagy that, has two membranes around it.
no, this microautophagy is inhibited by chloroquine?
By what?
Chloroquine.
Well, the microautophagy goes on during chloroquine treatment. What happens is that then the lysosomal products are not degraded. I actually showed a picture full of these little vesicles.
Sí,
So
creo
that's
que
the
tenemos
microautophagy.
¿Cuántos minutos? Well, I have one question. First of all, it's good to know that there is more to JSK than GlaxoSmithKline. In cancer cell, there seem to be many more vesicles moving in the cell than in normal cells, especially in cancers of the blood. And I was wondering if there is any connection between the Wnt pathway and the movement of those of vesicles, or even with cell proliferation in cancer cells.
Mm-hmm. Well, we haven't studied that specifically, and I didn't know that cancer cells have more vesicles, but what I showed is that you put Wnt and you get these huge vesicles, that these endosomes that just jump at you. Mm. And we have studied, one of the ones I showed there is called microophthalmia transcription factor, MITF, and there in MITF, we showed it's stabilized by Wnt, and when you stabilize by Wnt, just MITF, the stabilization of MITF by Wnt produces more lysosomes. It's a part of the family of TFEB, the transcription factor that makes more lysosomes. So you put more MITF, you make more lysosomes, you have more endocytosis, I think. So I would predict, and most people say, oh, Wnt, it's a very little bit of LRP6 that is on the cell surface and this and that, but you could see there these great big vesicles. So there must be a lot of LRP6, I would propose.
Thank you. There was a question somewhere here? No? Okay, Rafael. And
Eddie, I'm not sure if you mentioned this. What is doing the methylation doing at the structural level of the protein so that it becomes so different to the action of Wnt?
This which protein?
The methylation of the arginines.
The
What is doing at the structural level?
Well, I don't know. We're just working on this as we're now. But this previous work of my postdoc showed that it enhances the recognition by GSK3 a lot. So it makes a much more hydrophobic point, and so GSK3, and there are these methylated arginines are very close to the GSK3 site, so might easily help the landing of it. But in fact, now it's known there, I don't know, I think 20,000 or something, a lot, 10,000, let's say, methylated proteins, people that look at this. So many of them are methylated. And we have this other one called SMAD4, which I like to talk about because we have good antibodies. That one has the methylation site right in the middle of all the three GSK3 sites. And it's the only methylation that it has, so we know a little bit more. So it helps, we would imagine, GSK3 land in its right place.
Thank you. Juan Carlos.
Yes. I've been interested in muscle atrophy, and in denervation, 70% of the atrophy is justified by hemi channels expression, but 25% is autophagy. And in cancer, the muscle atrophy is indication of a bad prognosis. When somebody has a lot of atrophy, it means that will live very short time. And there is no hemi-channel involvement, but it's all autophagy. So maybe, as you mentioned, in all pathological conditions, autophagy is important. So the Wnt signal is very important, but the relative importance is different in different pathological conditions. So we have some pathological conditions like cancer and atrophy in cancer, muscle atrophy, maybe is 100% important, but in denervation it's probably only 25%. So, and it goes.
Yeah. Well, we should make the distinction between autophagy and microautophagy. Because autophagy has double membranes and it's stimulated by starvation, right? This is just part of the endocytosis process.
This is the
so it could have an effect or maybe not. Now, the big thing in muscle is the, what's it called? The SMAD4 pathway.
SMAD4,
that has TGF-beta that makes big muscle, the cows with Charolais, But anyway, I don't know about muscle atrophy. But let me just say, since we're just talking, that when this Wnt signaling, Christoph Niers has shown, is maximal by a lot more during the G2 phase of the cell cycle. And he has proposed that this is a mechanism which he called Wnt stop that will, when the cells are about to divide, they're going to stop the protein degradation and become larger so that they can now divide into two large cells. So this is probably related to the cell cycle, but this thing of the cell size is a very new kind of thing.
All right. Quick one.
Eddie, very, very nice talk. So in immunity, we know that the Wnt beta-catenin pathway is related to the generation of immunosuppressive dendritic cells. And you talk a lot about TGF-beta, but nobody knows why the Wnt beta-catenin pathway is related to tolerogenic immunosuppressive disease. But you talk about TGF-beta and SMADs. So probably, do you think it could be related to the lack of degradation of the TGF-beta SMAD pathway?
Well, I don't know anything about immunology, but we have a paper in Cell Reports 2015 showing that when you put Wnt, TGF-beta is active at, I don't know, much, much lower concentration. So it potentiates TGF-beta enormously, the Wnt. So